Hi, everyone. My name is Laura Beth Calvis. I'm a postdoctoral fellow at the University of Pennsylvania and recipient of the 2020 Founders Circle dissertation grant. And today I'm here to talk to you about that work, which looked at the relationship between sleep disruption and delirium in critically ill children. So the purpose of this study was to develop a measurement framework to illustrate the relationship between modifiable characteristics of the pediatric ICU environment, which included light, sound, and clinician caregiving patterns, sleep disruption, and delirium in young critically ill children. And so the first aim of the study was to describe the environment sleep and delirium children were experiencing in the pediatric ICU. The second aim was to look for relationships between these three variables. And the third aim was to evaluate the feasibility of the measurement framework that I had developed. So some key findings from this study, looking at the environment of the pediatric ICU, I found that children were experiencing low levels of daytime light and high levels of sound during both day and night hours. However, neither one of these variables was related to sleep or delirium. The environmental variable that was related was clinician caregiving patterns. So I found that an increased frequency and duration of clinician caregiving was associated with both sleep disruption and delirium. And then in addition to that, I found that kids with and without delirium did have differences in their sleep patterns. So kids with delirium during the night experienced more awakenings from sleep and also a lower sleep duration than children without delirium. As far as participation in MNRS, I think the best thing about MNRS for PhD students is the career advancement opportunity. So I've been able to present here at MNRS about 4 different times, I think I counted. And it's a great way to get recognized for your research as a student. So my first conference in 2018, I received a poster award. Of course, receiving my dissertation grant was a huge accomplishment. And then in 2022, when I came back to present my dissertation results, receiving the Distinguished Abstract Award. So those are all really great opportunities that I received at MNRS. And then
So for my dissertation work, I looked at neighborhood disorder and epigenetic regulation of stress pathways. So I looked at 6 genes, 6 glucocorticoid stress-related genes, in addition to neighborhood components as measured by survey data from the Ross neighborhood disorder scale. So we looked at measurements of the neighborhood disorder using survey data from the Ross neighborhood disorder scale and also a crime scale. And we measured psychological distress using the psychological general well-being scale. And we also measured DNA methylation using the Illumina 850K bead chip array. But what we did find when we looked at the differences between women with term birth and women with preterm birth was that we had differences in 2 genes that were at CPG locations that may be likely to support gene expression changes. So both CRH and NR3C1 gene were found to be differentially methylated. In addition, when we looked at the associations between perceived neighborhood disorder and DNA methylation, we also found 2 genes and the same genes that were associated with perceived neighborhood disorder, and also another gene that was associated with psychological distress. So as far as MNRS, I will continue to attend and present at MNRS meetings each year. I think that I would really like to, when I am mentoring students, to bring them to MNRS, because I had that, myself, and I had great mentors. Cindy Anderson and Carmen Jujescu made sure as a student that I attended MNRS very early on. And so that's what got me really excited about research. And I think it's so important to bring students in as soon as possible to be able to have that exposure and get that excitement to keep moving forward.
The purpose of my dissertation study was to explore the social networks of farm owner-operators and the social supports available to them for mitigating the negative health effects of occupational-related stress. The specific aims for my study were to assess occupational-related stress among farm owner-operators, describe existing resources for social support available to them in their social networks, and identify the composition of farm owner-operator social networks that they access to mitigate occupational-related stress. Now, this includes individuals, groups, organizations, events, and other resources, as well as the characteristics or attributes of those individuals. Now, the major findings from my dissertation include that farm owner-operators reported moderate or higher occupational-related stress, with levels varying by sex and production. Specifically, females and dairy farmers reported the highest levels of stress, and these were significantly higher than other respondents. The personal networks of farm owner-operators had fewer members than networks of a similar purpose, what are typically called trust networks, from studies of both farming and non-farming populations. Now, funding through MNRS, it's a dissertation grant, made this study possible, allowing me to distribute a mailed survey and follow up with farm owner-operators. And since completing this project, I've disseminated the findings through a number of national and international venues, including the 2021 MNRS conference, the National Rural Health Association annual conference in 2021. I was invited to, in 2022, to the International Conference on Agricultural Health and Safety. And finally, in 2023, the Spatial Social Network workshop that was hosted by the Georgia Institute of Technology.